Share my screen. Yep. And get this out of the way. Yeah, thank you. Thank you for the nice introduction and your kind invitation for me to present our work here. So today I will talk about the link between aging and the adaptive immune system. So let's start with aging first. It is the common factor for the most prevalent chronic diseases, and the incidence rate of most diseases increase linearly by age, but for dementia at around age 65, you see here, there is a spike. And age 65 happens to be around the same age where we see a dramatic increase in the ventricular volume of the brain due to white matter loss. And the white matter includes the regions that are rich in myelin, all these myelin-wrapped neurons, very essential for neuronal connectivity. But the molecular processes that underlie such white matter pathology are not very well understood. And to study this pathology, we established a single-cell RNA sequencing pipeline in our lab, where we microdissect gray and white matter. Oops, what? Some technical problems, unfortunately. Let's see with the PowerPoint. No? Okay. Okay, hopefully it's working now, Claire. Okay. So yeah, to study this pathology, we established this pipeline where we microdissect gray and white matter and use a optimized dissociation method. Then using plate-based and droplet-based sequencing technologies, we generate single-cell libraries and data sets. And using this pipeline, in our previous study, we looked at microglia and how microglial responses are altered in aging. And we identified 4 major clusters of microglia, one of which was white matter specific, hence the name white matter associated microglia. And to sum up this long story, just briefly, the WEM form nodules. In this formation, they are engaged in clearing up the myelin debris that is accumulated throughout aging. So white matter-associated microglia are an important component for the maintenance of white matter integrity in response to this myelin pathology. But a question still remains that what leads to this myelin degeneration in the first place? So since myelin is produced and maintained by oligodendrocytes, we thought maybe oligodendrocyte aging trajectories and expression profiles could give us some clues. So we did multiple single-cell RNA sequencing experiments using our pipeline and with the SMART-seq2 plate-based method and 10x, a droplet-based method. And for both data sets, in the end, after quality control and cell type annotation, we extract the oligodendrocytes and analyze them in greater detail. We identified 4 major populations of oligodendrocytes that I'm going to talk in detail today. So the first 2 clusters, the most abundant clusters, are the oligo 1 and 2. These clusters represent the heterogeneity in the resting oligodendrocyte state that is already known in the literature. The next one is the aging-related oligodendrocytes. We named them as such because they are greatly enriched in the aged animals. And this cluster was characterized by high expression of SERPINA3N and C4B, and such a expression profile has been associated with injury responses, especially in AD models. And last but not least, we identified a small yet novel population of oligodendrocytes. As you can see in red here, the IRO is small interferon-responsive oligodendrocyte cluster. And this type of a subpopulation of oligodendrocyte has not been reported before. So there are some OPC populations with a similar transcriptional signature in EAE mice models, but not in oligodendrocytes. So this was a interesting hook for us to follow through. And the overall transcriptional signature in the aged oligodendrocytes was quite consistent between the two technologies that we used, with the IRO showing high expression of interferon response genes as well as MHC class 1 genes. But this consistency was a good indicator of reproducibility, but any bioinformatics or computational finding is never complete without validation experiments. So we did some validation experiments to see if these oligodendrocyte states are reproducible or not. And indeed, after co-staining with the ARO and IRO markers, the aging-related and interferon-responsive oligodendrocyte markers, we saw that both states are almost exclusively found in the old white matter compared to the young. And the next question was that what is inducing this interferon response in oligodendrocytes? And previous work has shown that the T cells are present in the aging brain, close to neurogenic niches and within the optic nerve, respectively, in these two studies. So we looked at CD8 T cells in young and old mice in gray and white matter. And what we saw was that the T cells were mostly CD8 T cells and almost exclusively found in the white matter. Then we wanted to study the spatial relationship of these interferon-responsive oligodendrocytes together with the CD8 T cells. And I just showed you the interferon-responsive oligodendrocytes are a very small, a rare population. And CD8 T cells in the aging brain are also a very rare population. But in the end, it turned out that these two rare populations are actually found in close proximity to each other, so indicating a potential interaction between them. But we wanted to explore further this connection between T cells and the white matter aging. So we performed experiments using rag knockout mice. So rag knockout mice are a very nice avenue to study the effects of T cells because these animals don't possess any mature B cells or T cells. So in the end, we integrated our wild type dataset with the rag knockout dataset that we have and ended up with almost 45,000 cells. And as before, we extract the oligodendrocytes and analyze them in greater detail. We identified the same 4 major clusters of oligodendrocytes, and we saw that the interferon-responsive cluster was greatly reduced in the REG1 knockout animals. And we did the validation experiments with some immunolabeling and saw that indeed the IRO numbers were decreased. So this showed that the interferon-responsive oligodendrocyte conversion is actually dependent on the presence of T cells. Well, one major point of contention when it comes to the rag knockout results is that these animals lack various populations of adaptive immune cells, not only CD8 T cells, but they don't have any B cells, CD4 T cells, natural killer T cells, and so on. So we used a CD8 knockout model so that we can investigate specifically the effects of absence of CD8 T cells. And we again see the reduced number of STAT1 positive oligodendrocytes in the CD8 knockout model, replicating our initial finding. And up to this point, I showed you results focused on oligodendrocytes and how the adaptive immune system mediates changes in oligodendrocyte states in aging. But previous work shown that there is a interferon-responsive microglia state in disease models and as well as in the aging context. So we asked the question, does the adaptive immune system mediate interferon-responsive microglia conversion as well? And we had the ideal data set in our hands to answer this question using our integrated data set. This time we extract the subset the microglia population and analyze it in greater detail. And unbiased clustering led us to find these 4 clusters that we previously described in our paper, in the previous paper, as well as a smaller interferon-responsive microglia population. And this interferon-responsive microglia cluster was significantly decreased in the REG1 knockout white matter compared to his wild-type counterpart. And we again did the validation experiments and saw that at 3 months, this STAT1 positive interferon-responsive microglia cluster was almost absent, and it was significantly reduced in the old REG1 knockout mice. Also, we replicated this finding with the CD8 knockout model, and similar to the oligodendrocyte proximity analysis, we looked whether the CD8 T cells and the interferon-responsive microglia co-localize to each other, which indeed turned out to be the case. So with the REG knockout or CD8 knockout model, we looked at how the interferon-responsive cell states are affected when the adaptive immune system is suppressed, when there are no lymphocytes, there are no CD8 T cells. So we wondered what would happen if we tipped the balance to the other way. So we conducted some immune checkpoint blockade experiments using antibodies against inhibitory molecules, CTLA4 and PD1, essentially blocking the inhibition of the immune response, so promoting the T cell activation even further. And this led to a several-fold increase in the number of CD8 T cells, as well as in the number of interferon-responsive oligodendrocytes and microglia. So rather than just correlation with the proximity analysis, at this stage, we built up a causal link between CD8 T cells and the interferon-responsive oligodendrocytes and microglia. And we did this from 2 angles. First, while looking at the changes in the absence of lymphocytes, and then while looking at what happens when the T cell response is actually enhanced. And taken together now, the data I've shown you are evidence that the adaptive immune system is a driver of interferon-responsive states in oligodendrocytes or microglia. But it is still not so clear to what extent these changes are contributing to white matter degeneration. So we quantified oligodendrocyte numbers in 12, 18, 24 months wild-type animals, as well as 24 months REG knockout animals. And we observed no differences in gray matter density between the experimental groups. There was a gradual decrease in white matter density starting from 12 months to 24 months old. But strikingly, this loss of white matter effect was rescued in the REG knockout animal. So this was a very exciting result for us, which basically means that by eliminating T cells, we were able to prevent a major effect of aging, which is white matter loss. So the connection between neurodegeneration, adaptive immune system, aging seems to be quite complex and multifaceted and very worthwhile to investigate. And the nice thing is that from a neuroimmunological perspective, there are already so many drugs in the immunological compartment, so many established, tested, validated drugs that target CD8 T cell biology, like blocking CD8 T cell activation in autoimmunity or autoinflammation conditions, or just directly interfering with the interferon pathway via JAK kinase signaling, Janus kinase signaling, and so on. So the targetable immunological arsenal is quite large when you compare it to the neuronal compartment. So we have a lot of already studied targets to take care of neurodegeneration. So I talked about the link between neurodegeneration and CD8 T cells in the context of aging, but recent studies are telling a similar story in different conditions like ALS and AD. And I don't want to give any spoilers for the next talk for the AD parts, so, but it's just a very reassuring, it's a good feeling when a well done and well detailed study reaches similar conditions in parallel to yours in different contexts. So yes. So overall, I think the role of the adaptive immune system is a untapped source of new findings and information to study various aspects of neurological conditions. And in response to this, we are seeing more and more publications every day studying T cells, connection to neurodegeneration in different contexts. So the future looks to be very exciting and full of new discoveries. And with that, I'd like to thank my advisors and my fellow lab mates who contributed to this study. And I'm happy to take any questions. And thank you.
That was a really fantastic talk. That was very interesting. So thanks for sharing that. And now if anyone has any questions, feel free to unmute yourself and ask, or if you want to put something in the chat, I'm happy to read it out as well. Take it away.
May I ask something? So what do you think now it will be the role, if I don't know, if a stress or different lifestyle into white matter aging? Do you think there might be any correlation to make it worse?
Yeah, that's I mean, that's coming from a point of individual variability, kind of stress and different factors. That's one of the problems to study, is the role of adaptive immune system when it comes to the brain. And because these factors that are not easily controllable and that are not easily reproduced is showing, and then the cells are conditioned against different antigens based on this hard to replicate, hard to reproduce parameters. So it's in the end, we have a very uncertain type of, it's all speculation. I don't think there's any empirical evidence to say something in one direction. But yeah, to speculate, I would think so. The stress and the inflammation throughout aging is a very important proponent of the white matter degeneration, but this is pure speculation. Like I say, it's very hard to control and generate empirical data on this.
Thank you.
Thank you for the question.
I have another question for you here, from Anne Marchildon. They're asking, can you isolate the IFN gamma response is due to microglia versus astrocytes?
Okay. I'm gonna share my screen back. So for the interferon gamma responses of oligodendrocytes, if I can find my backup slides here. Where is it? Okay. So what we did was that we were also curious about the effects of interferon gamma, and we just treated the oligodendrocyte cultures with interferon gamma. This is what you're seeing here. And in the end, we were able to induce this interferon-responsive oligodendrocyte state, but there were no changes in the number of oligodendrocytes. But then we co-cultured microglia and oligodendrocytes. Then we did the interferon gamma treatment, and it was carnage. Basically, we lost quite a lot of oligos when we just co-cultured them together with the microglia. So indeed, microglia is leading the charge here, because without the microglia's presence, we don't see the oligodendrocyte loss. But when the microglia are in the picture, we see a great loss of oligodendrocytes. But when it comes to astrocytes, I'm not so sure. We don't have data on that. But in vitro, at least, microglia is a important factor in this pathology or loss of oligodendrocytes. But astrocytes, yeah, I don't have any data on that. If hopefully answers the
I
question.
have a follow-up question, kind of based off of this. I was wondering if you've tried to inhibit IFN gamma in any way, and if you have, did that kind of stop this cascade that you described?
Yeah, that's a good question. That's kind of a follow-up project type of thing. So no data is available at the moment, but yeah, it's a good question to follow through. Indeed. Yeah, interferon gamma is putting microglia in such a cytotoxic state that it's eating up the oligos, or is leading to this oligodendrocyte death in the end. But in vivo, this could be, of course, this could be due to directly CD8 T cell cytotoxicity or other factors. Maybe even astrocytes are playing a role directly or indirectly, or other cell types. And yeah.
Great. I look forward to hearing how that goes.
Thank you.
Any other questions?
Yeah, maybe I could ask a question. I guess you've addressed my previous questions, but this interferon gamma is really interesting to me. So first of all, that's a really fantastic talk and really fantastic data. Very well done. I guess I'm intrigued at the differences between the different interferons. Like often with Alzheimer's disease, you'll get a type 1 interferon response.
Yeah.
Both with your work and with our work, it seems like interferon gamma is the more toxic element. I'm wondering what's special with interferon gamma. Is it basically the interaction with the T cells, or is there something other to it?
Yeah, that's also another great question that unfortunately I don't have the answers, but it's also something we're looking in this type 2 versus type 1 versus type 2 interferon signaling, how it's affecting. There is some evidence the interferon beta is blocking the neural stem cell proliferation, but yeah, gamma is doing the same. But yeah, we're not sure about this. Also something that we're looking into, but yeah, I don't have any data, unfortunately.
Yeah.
But it's a
in
great...
COVID, actually, interferon gamma seems to be a lot more toxic than the other interferons. I realize it's not brain tissue, it's lung tissue that it's ultimately destroying. But again, it's a macrophage interaction with interferon gamma that seems to cause the lung damage and severe COVID. Also, Shane Liddellow's work talks about a toxic trio of cytokines, and interferon gamma is one of those with TNF alpha and IL-1B as well. So there seems to be something particular and interesting about interferon gamma that's more neurotoxic. And yeah, I guess I find that interesting.
Yeah, seems to be that way. And one kind of limiting factor is that it's hard to study the type 1 versus type 2 changes in the transcriptomics sense. So it's all interferon response, interferon stimulated genes and so on. But yeah, that's why it's kind of hard to look from this perspective. But yeah, very, very good point. We're also
we've actually tried to do that in culture, and we see that there's an overlap with all of them. Like some of them are more potent than others, so that's probably something to do with it. But yeah, it's very difficult to study.
Yeah, exactly. Okay.
All right. Well, thank you again. That was very, very interesting, and just a fantastic presentation. Yeah.